Where does it happen? In the cristae of the mitochondria. What is this thing called the electron transport chain? Well, it's just a number of proteins embedded in that innermost membrane. Let's go ahead here, maybe. Okay. Here it is. Just a number of proteins embedded in the pathway, or I'm sorry, embedded in the membrane. Okay. And I mean, they're labeled pumps. Okay. But I mean, they are proteins. Okay. They help to accept these electrons. Okay. Here's electrons. Okay. NADH brings electrons here, dumps them off. Okay. FADH2 brings electrons here and dumps them off. Remember, these electrons, they carry energy within them. They get passed, these electrons, from one carrier to the next, sucking all of the energy out of it. We suck it out of that. We suck it out of it till ultimately there's nothing left. Where am I? There we go. Okay. That is the electron transport chain. I'm transporting these electrons from one carrier to the next, harnessing that energy from those electrons. How does it harness that energy? Well, it harnesses that energy that's within those electrons, and as you can see, uses that to create this thing called a concentration gradient, right? It's taking the energy in those electrons and using it to pump hydrogens across as it does. It's how we utilize that energy in those tiny little ions, or those tiny little building blocks of those ions. As it comes across here, again, into the second pump. And as it goes from one side to the other, past to this carrier to the third pump, I suck more energy out of it, pump more hydrogens out. And then the third pump, my third chance to take whatever life I can out of that sucker and utilize it, pump hydrogens out. Well, now I have all these pools of electrons sitting here with no energy to speak of. We don't want that. I want to get rid of those SOBs. I want them out of here. So now what do I do? Well, I have some hydrogens that are just floating around in here, right? Okay. Okay. Obviously, I've been pumping them out. I can take a couple of those suckers. Again, oxygen is present. And I can utilize those electrons to get hydrogen ions to bind with oxygen and form water. Remember, the complete breakdown of glucose ultimately forms CO2, which we've seen, and here's water. Okay, this is metabolic water. Metabolic water is water that's formed during the process of metabolism, cellular respiration here. And we can remove it. Remove it from the cell, remove it from the system. As I've sucked all the energy out of these electrons, as I've passed them from one carrier to the next, and these are the names of the carriers here. I'm not going to ask you, okay? These are my carriers. There's some cytochromes, all right? But these carriers accept those electrons, okay? And as we accept these electrons from here and pass them from one carrier to the next, I suck the energy out of them. Suck the energy within them and pump out hydrogens in the process. And it creates a concentration gradient. I have a large number of hydrogen ions over here compared to over here. So now I can get these hydrogen ions here to come down their concentration gradient. And just like here, where I use the electron to pump hydrogen across, now as it goes down its concentration gradient, what can I utilize? All the power within that hydrogen ion to add an inorganic phosphate to adenosine diphosphate. And I can make several units of ATP. You may have heard to that be called chemiosmosis. Okay. We form a chemical gradient, and we use that chemical gradient to fund the ability to make ATP. What enters, or where does this occur? It occurs in the cristae of the mitochondria. What enters? NADH and FADH2. What exits? Water and energy, I guess, right? Okay. But water is the massive thing that exits here. And water doesn't have any energy within it. That's what all of these slides are getting across. Okay. Phosphorylated, whenever you see this word, what does it mean? What are you adding? What are we adding?
A phosphate.
Phosphate group.
Yep. Thank you, boys. Adding a phosphate. Okay. That's all that means. Is it 100% effective? Of course not. Nothing in the body is. Nothing in the body is effective. 100% effective. Okay. This is like 33% efficient. All the rest of that remainder has just lost its heat. Lactic acid pathway, it's only 2% efficient. Okay. There's a huge waste. It's not necessarily a waste. It just takes a lot of energy to to pump out lactic acid out of a cell, transport it to the liver, convert it into glucose, transport it back. So that takes a lot of effort to do. Okay. So that's why it's only 2% effective. All right. There you go. Oxygen, final electron acceptor. I mean, that's probably it. Okay, that's probably it. I am not going to ask you to distinguish between a theoretical yield and an actual yield. How much do you get from that electron transport chain? Well, that's what I have circled right here. Okay. Get around 26 or 28 ATP depending upon the cell. Prior to that, we've only gotten 4 ATPs. 2 of those came from the very first step. The other 2 came out of the citric acid cycle. Okay. So this big yielder of energy is going to be the ETC. Where do they occur? Who enters? What leaves? What are you getting out of it? Okay. Glycolysis, cytoplasm. Everybody else is happening at someplace inside the mitochondria. Matrix, folds, the cristae. Okay. Who enters at each step? And what do you get out of it? Where do they occur? Those are always things that I generally ask. And I usually ask it as multiple choice, if you must know.
Because you're going to get ATP out of every one of these steps. It's just varying amounts and then some quote unquote like byproduct with like it could be water in the case of the electron transport chain or citric acid in Krebs cycle.
Yes.
And glycolysis is comes out of glycolysis.
Just pyruvate.
That's right, pyruvate.
Yep. Pyruvate. And a couple ATP and a couple NADHs. Okay. That is what I ask about that. And again, I usually do it as sometimes I do it as a short answer, but lately I've been just doing it as a multiple choice question.